Well, good afternoon, everyone. It's a tremendous pleasure to be here. And I'd like to just start out by telling you a little bit about the origins of this conference. To my knowledge, I think it came about initially due to a conversation that Keith Gillis, who's the Dean of the College of Natural Resources here at UC Berkeley, had with Neil Gutterson at DuPont Pioneer about the possibility of hosting a conference that would invite a public discussion around the ethics of genome editing. And we've been delighted here at Berkeley to be able to host this meeting. A lot of thanks go to many people who have been involved in organizing it. That's certainly not been me, I have to say, but Melody Cantarutti, Diane Ferris, Susan Jenkins, and the team at both the IGI and QB3 have been tremendous, as well as the professional organization that are behind putting this all together. So huge thanks to them. And also thanks to all of you for your interest in this issue and coming to Berkeley for a couple of days of, we hope, very exciting and very interesting conversations. So the goal of this meeting is really to stimulate input, and I think you can see from the great introduction by Michael Krasny that we have a number of venues for you to do that. I thought what I would do in my introductory remarks today is really just to tell you a little bit about the origins of gene editing from the perspective of a biochemist like me, and how I came to the realization that this was going to be an area that would have profound implications not only for scientists like me, but also for everyone else, because of its potential to impact human societies in different ways. And so I wanted to really just start off. I know many of you are not necessarily scientists by training, so I thought I would just start off by giving you a very short introduction to the background to CRISPR-based gene editing, what it is and what it enables scientists to do, and then talk about some of the ongoing applications of gene editing that will, I hope, show you why there's so much excitement about this and what the potential is for the near future and also in the longer term. And then I'd like to, at the end, get to some of the, I think, really pressing ethical questions that we all now face with this powerful technology, and how we'd like to be able to engage together in thinking about responsible ways to move forward. So let me just start by pointing out that we can think about the way that cells and organisms are put together by understanding that the code of life is really is the DNA inside cells that provides all of the information telling a cell or a tissue or an organism what to do. And you can think about it a little bit like you might think about the text of a document, for example, that has instructions or an instruction manual. And for that text to make sense, the words and sentences and paragraphs have to come together and have to be read and acted upon. And so when we think about gene editing, what we're really talking about is a way of modifying that document, if you will, the DNA inside of a cell. We can think about it like, what if you could move sections of text around, you could change individual letters in that document, you could cut and paste text, and that's, up until a few years ago, was really kind of a fantastical idea. It sounded very science fiction-y, it still sort of does, but today there are technologies available now to be able to do this in a very precise and predictable fashion. And this gives scientists who want to manipulate organisms, whether it's everything from mice to plants to bacteria to even human beings, a very powerful way to manipulate the set of instructions at the level of the DNA chemistry of that organism and control the way that that information is used and what it says and what it tells the organism to do. And I think it's interesting to point out that the idea for doing this kind of manipulation in cells is not new. In fact, I would argue that it really stems from the 1950s, when scientists like Jim Watson and Francis Crick and Rosalind Franklin and others discovered the structure of DNA, the double helical structure, and that already gave scientists the idea that you could understand the code of life, and by understanding it, figure out eventually how to manipulate it. And nobody at the time knew how that might be eventually possible. But over the years, various technologies have come along for what we call gene editing, namely being able to precisely manipulate the DNA inside of cells. And there have been a number of iterations of this, starting with, I would say, sort of chemical methods of doing this, up to a few years ago, methods that involved programmable enzymes, being able to engineer proteins that could find and change particular DNA sequences inside of cells. And then, a few years ago, scientists who were studying something seemingly unrelated to gene editing, namely how bacteria fight viral infection, came across a system known as CRISPR, which is an adaptive immune system in bacteria, a way that bacteria can find and destroy viruses by recognizing virus DNA in bacterial cells. And it was through the understanding of how that system operates that it was possible to harness the enzymes, the proteins that are part of this bacterial immune system, as a technology for gene editing. And I'm not going to get down into the weeds on how that works, but I want to tell you very briefly what this technology enables and what it does. And it really came about through investigating the way viruses infect bacterial cells and how those cells respond. And so what I'm showing you here is a picture of a virus that's landed on the surface of a bacterial cell, and it's in the process of infecting that cell. And the way infection works is the DNA from the virus is literally injected into the cell, and this bacterial cell then has very little time to detect the virus DNA before the virus is able to make many copies of itself and destroy the bacterium. And so in those bacteria that have a system called CRISPR, they can recognize this injected DNA as foreign, and they have a system of programming proteins with little bits of molecules called RNA. These are copies of virus DNA that can allow the cell to detect and destroy virus DNA in a selective way. And very briefly, just for those of you that care about the sort of underlying molecular basis for this, the way it works is by the function of a protein. I'm showing you here in this picture a 3D printed model of the protein called Cas9 that is an RNA-guided enzyme. And what does that mean? Well, it means that this protein grabs onto a piece of RNA, which is the orange molecule in this model, and that RNA molecule has a sequence of letters in its code that is able to match the sequence of letters in a piece of viral DNA. And when that match occurs, this protein with its guiding RNA holds on to the DNA, which is the blue double helical molecule in the model, unwinds the DNA and allows the protein to function like a pair of scissors. It literally cuts the DNA strands and allows the cell, if it's a bacterium, to destroy the cleaved DNA. Now, it turns out that in cells like our own, human cells, plant cells, other kinds of cells of complex organisms, the cells have a sophisticated machinery for repairing DNA that gets broken in this way. And so when DNA is cut, it can be altered and in other words, edited at the site of the double-stranded DNA break. And so the really exciting power of this technology is that this protein, Cas9, is a programmable enzyme that can be programmed chemically to find and cut any desired DNA sequence in a cell, and thereby triggering a change to be made in the DNA at that position. And that's really fundamentally how this operates. And I was going to just show you this, which indicates that when a DNA molecule inside an animal or plant cell receives a double-stranded break, like in this cartoon, the cell can repair that break either by making a very small change to the DNA in the process of repairing the break, which typically leads to disruption of a gene, for example. Or scientists can introduce a piece of DNA that gets incorporated into the broken segment of DNA during the process of repair. And by doing that, we can actually incorporate a new genetic information at the site of the DNA break. So this is a tool, in other words, that can be used to knock out genes or disrupt, remove genes from the instruction set in a cell, or to introduce new genes or new genetic information into a cell. So it really goes both ways. Now you saw this wonderful video, so I'm not going to show you this again at the beginning, but this really is a system that remarkably operates in effectively every type of cell where it's been tested. So I wanted to give you, just to give you a sense of just the pace of research in this field, for those of you that aren't reading the literature every week, scientific literature. These are 3 applications of CRISPR technology that have come out just in the last few weeks. So one idea that's being explored actively is, can CRISPR be used to treat people that have a viral infection in which the virus has actually integrated its genetic information into the genome of a cell. And so this is an example in doing experiments in mice that have a disease sort of mimicking the HIV infection in people that causes AIDS, and asking whether the CRISPR system can be used to remove the integrated piece of viral DNA from the mouse genome. And in fact, this experiment showed that that can work very well. So the idea here is, to think about using the CRISPR system in a way that's almost analogous to how it's employed in its native host bacteria. Another really interesting example from very recent work comes from a group at Cold Spring Harbor Lab that was able to use the CRISPR technology to alter tomatoes so that they would be much more capable of bearing heavy fruit yields. And this was done by employing the gene editing technology to do something that traditional plant breeding would have been, extremely difficult, if not impossible, to do. And so one of the reasons that this got some press in the media and was on the cover of a prominent scientific journal is that many scientists think that in the future it will be possible to deploy the CRISPR technology very broadly in different types of plants, not just the most famous and widespread crop plants, but also in other kinds of plants that occupy various niches, both commercially and in people's gardens, to enable plants to be generated that meet various needs in just very localized niches. And then finally, you might have seen this picture recently. Work that's been ongoing in the George Church Lab and now by a company that he's started and others is aimed at generating animals that are going to be better organ donors than what we've had in the past. And so this is a very recent work that was published using the CRISPR system to remove viruses from that are sort of naturally part of the pig genome, and then creating pigs that are virus-free. So these are animals that in principle would produce organs that could be more applicable for organ donation because they don't have the ability to spread endogenous viruses that are present in most pigs. So in my own sort of research, you sort of have to imagine that, I'm a biochemist, and I've always studied molecules and done work on fundamental molecular mechanisms. And when we did the work that we published back in 2012 with our international collaborator Emmanuel Charpentier, and was work that was actually started from conversations I had with Gillian Banfield and her group here at UC Berkeley, it was not on our radar at the time how this technology would become such a powerful tool across virtually every area of biology. But it quickly became clear that scientists were adapting this very quickly for gene editing applications across a very broad spectrum of organisms and cell types. And one of those applications was to make modifications to what we call the germline. So that means being able to make changes to DNA in cells that, and doing this in the context of a fertilized egg, in other words, an embryo, such that the resulting genetic changes become part of the entire organism. And not only that, they can be passed on to future generations. And this is an example just showing you this is a mouse embryo that's being held by a pipette tip coming in from the left, and you can see a needle entering from the right and injecting DNA editing molecules into this embryo. And it turns out that that kind of application of CRISPR technology works very robustly in mice, rats, many other types of organisms, including in mammals. And I wanted to show you, I'm going to show you a couple of pictures, the very nice experiment that I learned about from a student, Sonata Kodka, who's a student in Texas, who was doing experiments using germline editing to modify frog embryos. This just illustrates how this actually works. So this is an experiment in which Sonata had a collection of 2-cell embryos. These are frog embryos. And the experiment here was to inject one of the 2 cells with the gene editing molecules along with a gene that encodes a green protein. And you can see that in every embryo that received this injection in one of its 2 cells, the cell that got injected has now a copy of the gene encoding the green protein, and we know that because the cell turns green. And these embryos are perfectly healthy. They can be cultured and allowed to develop into tadpoles. And when you do that, what you find is that these animals have half of the cells in the organism are expressing the green protein. We can see that when we put the animal under a fluorescent light, and the other half of the cells are not. And these animals are otherwise completely normal. If you sequence the DNA from these cells, animals' cells, you find that the change introduced in the DNA is precisely at the position where the gene editing molecule made a cut and allowed integration of the green protein encoding gene. So it's really a remarkable tool. It just shows you kind of the specificity and the precision and the power of a technology like this. So early in 2014, a research lab published a paper using this kind of strategy of germline editing in monkeys. And I found out about this because I was sitting at my desk upstairs in this building, and I got a call from a reporter saying, what do you think about this work that's being published this week on monkey editing? And so I got a hold of the publication, and it really had kind of a profound effect on me, because I started thinking about this, and the thing that was so amazing about the work that was in this publication is that just like I showed you in the frog embryos, these scientists were able to make a precise change in a single gene in monkey embryos that had no effect otherwise on these animals. They were able to develop into normal monkeys, but when their DNA was sequenced, they had a precise change in one gene of every cell of the animal, and these changes were a part of the germline now. In other words, they could be transmitted to future generations of monkeys. And I started thinking about this, and I thought, well, if you can do this in mice and rats and monkeys, why not humans? And maybe there are people out there that are already doing that. And it was sort of for me at least, kind of a really arresting thought. And I got together with colleagues here at Berkeley and elsewhere who were likewise thinking about the impacts of gene editing. And this was really the beginning of conversations about how we think about the effect of a powerful technology like this on society, and how we grapple with the responsibilities that this technology brings along with it. And that led us to sponsor a meeting in January of 2015 that was attended by, among others, David Baltimore and Paul Berg, 2 very illustrious scientists, both of them Nobel laureates, who were involved in the conversations in the 1970s about the ethics of molecular cloning. And so they were very helpful in helping to frame the discussion around gene editing, in particular its applications to human beings and applications in the human germline. And that meeting then led to conversations about how sort of leading up to eventually meetings like this, thinking about how we would engage with a much broader cohort of people, explain what this technology was all about, and how we could all talk and discuss together about how to move forward in a responsible way. So of course, there's been lots of interest in this, and this is actually a photograph taken from the cover of The Economist magazine from last year, under the title, the banner, Editing Humanity. And there's been much discussion about the potential to modify the human germline, culminating, at least most recently, in publication, I think it was just last week, by a group up in Oregon who were using viable human embryos to make modifications, and doing so in a way that effectively presents a potential protocol for a future clinical application. And so, we can sort of see this, the sort of inexorable progress of this kind of application. I think there's a lot of interest in it, but clearly many important and fundamental questions that need to be addressed about access, about, of course, the ethical and societal implications of this kind of application of gene editing, and many other questions that come along with it. So I'm sure we'll get into that as over the course of this conference. I do want to point out that one of the outcomes of that January 2015 a very small meeting about gene editing was to motivate a much larger conference that was held at the end of 2015, sponsored by the national academies in the US, China, and the United Kingdom. That was an international meeting. Some of you were there, and the discussion focused around human genome editing, and in particular, human germline editing, and how to think about the governance of that. And I urge you, if you're interested in that topic, to take a look at this report. It was released in February, and it provides a very careful analysis by a group of scientists about the potential of the science, and also what the implications of it are, and recommendations about how to proceed in the future with regard to applications of this type. So I thought I would just end by posing a few questions to you, things that I'm thinking about and that I hope will be touched upon in this meeting in the context of gene editing in various different kinds of systems and organisms, and for various commercial applications. I think we need to think a lot about the timeline, what needs to happen for gene editing to be widely adopted by technical practitioners. Now, this is of course happening in developed parts of the world, but what about in developing countries? I think we need to think about how gene editing is going to be democratized even further in the future, and how we can encourage that to happen in ways that, again, are responsible. I think we have to really consider the cost considerations for developing genome-edited products from the perspective not only of the developers, but also people that are the end beneficiaries, and then those that are in between. And I'm amazed at how many people in the agricultural community have reached out, especially recently, just over the last month. I've had a number of contacts from groups of farmers in the Midwest who are realizing that this technology is going to be impacting them in the future. They want to understand the science, they want to understand the implications. So I think this is something that we'll be grappling with on many levels and in many different types of applications of gene editing. And then there's the issue of social acceptance, and I think that was certainly one of the original motivations for holding this conference, is to think together about how we imagine people reacting to recognition that gene-edited products are now coming to market. So, sort of a classical example of this is golden rice, which has had a very, tortured history in a way. And, can we look at the way that these kinds of products have been handled in the past, and learn from both the mistakes and the successes of those products in how we think about presenting this kind of science and the resulting products coming from technologies involving gene editing, and presenting that to the public. And this is it's a very challenging issue, and I think that for many people I think that just understanding the science going on that's sort of involved here is a challenge, and then trying to understand how that's going to impact them, their health. This is something that we really have to grapple with, how to do it effectively. And I don't think there are easy answers, but I think starting with conferences like this is a good step in the right direction. And then finally, thinking about ethics, are there safeguards that guard against various kinds of side effects of genetic manipulation? There's been a lot of discussion in the media about things like what are called off-target editing, which could refer to changes made to DNA that are undesired, or we can also define them as changes made to DNA at a desired place, but that have undesired or unintended consequences. So this is a big sort of scientific issue, but of course it also has ethical implications, especially when we think about using these applications that occur in humans. Are there safeguards for agricultural use that avoid unintentional dissemination? Big question also for gene drives that affect insects and organisms that are released in the environment. And I think that scientists really need to take a very active role, not in dictating what should happen, but certainly in engaging in this conversation. And I was very delighted to see in the answer to that, one of the questions posed to the audience that Michael Krasny put up earlier, that many people here obviously feel that that's very important, that scientists need to be engaged in this discussion actively. And it's always a challenge to think about how we explain technical work in a non-technical or accessible fashion. But I think that's it's a very good goal. It's a very important thing that we all need to grapple with. And that's really where I wanted to stop. And I just want to thank a huge number of people that have put together this conference, the scientists and researchers here at Berkeley and across the globe that we've had the great pleasure and honor to be able to work with, and to invite you to really engage over the next couple of days. I look forward to the discussions, and I think I'll learn at least as much from all of you as you're going to learn from the scientists. So thanks again for coming. And I don't know if do we have time for questions, or how would you like to do that?
We'll have time for a few questions. If you do, once again, thank you for that wonderful overview. So the questions are in order, and thank you and the audience for presenting them. First question that kind of gets top billing is, how can we ensure policymakers are educated in science to make good decisions for society?
Well, that's a really important question. I think we have to just, we have to reach out in every way that we can. I think it starts with all of us talking to our families and our communities about what we do and our thoughts about these types of technologies and applications. But I think for any of us that are interested in doing it, there are many opportunities to go to Washington, to talk to our congressional representatives. In my experience, they've been very interested in trying to learn about the science, so I think that's something that we all have to take an active role in doing. So I don't think there's one easy answer. I think it's going to be a continuum of reaching out at any level you feel you have time and ability to do.
There may be no easy answer to a few more questions, then we'll go out in the audience, give you an opportunity to pose questions from where you're seated. What does the public have wrong about genome editing?
What do they have wrong about genome editing? I think it's important to appreciate what it can and cannot do. So it's a wonderful technology. It's not a magical technology, right? It's not perfect, and there still are a number of aspects of just the technology itself that are very much at the bleeding edge of science and are under active development. But I think it's also equally important for the public to appreciate that this is an incredibly fast-moving area. I mean, I've been doing science for almost 25 years professionally, and I've never seen, science sort of move at the pace that it's moving right now. So I think any of us that are involved actively in the research involving gene editing appreciate that. So I think it's something that the public needs to understand, because it means that we can't sort of put off these conversations, right? We need to all be really actively thinking now about the implications that are going to be facing us in the very near term.
Thank again the audience for these good questions. Let me one more, and I think it's really pertinent. How do we ensure that policymakers are educated in science to make good decisions to society or for society?
Yeah, I think that's very much aligned with the earlier question. I think we just have, I think all of us that have the ability and the time and the interest need to do our part to reach out to explain what it is that we're doing, our understanding of the science and the technology and the implications of it, and do that at any level you feel comfortable.
And maybe one more, how do we overcome the anti-science bias that's becoming such a part of the political discourse?
Yeah, this is something I think about a lot, and I don't think there's any easy answer, but I think, by, at least by showing that we're willing to, as scientists at least, to engage in these conversations, to take the time to explain what we do, take the time to learn how to talk about our science in a non-technical way, these are all important steps in the right direction.
Maybe we'll go to the audience now. If you have a question and you'd like to pose to Jennifer, just raise your hand and we will go to you. Otherwise, I'll go to the questions that have come from your devices. Over here. Jennifer, over here. Please stand up. Yeah, as cogent as you can be.
Some of the genes act alone, they will interact or static interaction. So if you start gene editing, you can't always predict how these interactions how do we actually do this in a sensible way?
Right, so he's pointing out that genes seldom act alone, they act really in concert or in pathways, and how do we understand the effects of editing one gene and how that might impact whole pathways or basically have unintended consequences, I think, right, is ultimately what you're asking. And, again, I think it's going to come down to a combination of doing careful research, understanding better what the interactions are, what gene networks exist in cells, and frankly, CRISPR-type research tools are already helping to do that. But it's going to be an ongoing question of trying to ensure that edits that might be made to an organism don't have unintended consequences. And I think it'll be important to put proper controls in place and do the right experiments to validate that.
One more question from the audience. Just raise your hand, we'll come to you. Anybody else from the audience who would like to pose a question? Yes, over here.
Would you suggest that we determine which genes are appropriate for germline modification? It seems like such a gray area. You can have monogenic diseases that are very clear and then just a myriad of other polygenic conditions. So how do you make that distinction of when it's appropriate, would you say?
Yeah, so that's certainly a question that could be debated and discussed for the entire time of this conference if we wanted to. But my short answer is that I think that, and if you read the report from the National Academies, the recommendation there is really that we look for situations where there would really be no other reasonable way to deal with a genetic disease other than gene editing. And when you really think about it that way, those situations are rather rare, right? And the reason is that already with genetic counseling, with pre-implantation genetic diagnosis that can be done during in vitro fertilization, there are ways to remove embryos that would have a deleterious gene in those ways that don't involve gene editing. And I think that in most cases that would be sufficient to deal with a monogenic disorder. But it gets more complicated when you have, if you have two parents that both have the same gene that's affected that causes disease. That's an issue where gene editing could be relevant in the future. And then you get into situations where people might start to say, well, if gene editing works very well, and we trust the outcome in embryos, maybe we want to put genes into embryos or make modifications to genes that provide healthful outcomes. For example, reducing cardiovascular disease or susceptibilities to dementia, things like that. Gets into a very gray area of are we talking about health or are we talking about enhancement? So I hope we'll get into some of those discussions later in the meeting, because I think these are now questions that we all have to grapple with given where the technology is today. So it's been a very interesting few months with respect to human embryo editing, because what's happened is that there were a series of publications that came out of Asia that focused on making gene edits to first non-viable and then eventually to viable human embryos. And those publications, although attracting some attention, were not so interesting. They were not so maybe informative to scientists because they didn't really present a comprehensive protocol for how you might actually use gene editing in that type of system. But more recently, just in the past week, there was a publication that came out that really, I think, did present a pretty comprehensive protocol for how you could use gene editing in viable human embryos to correct a genetic mutation. And that article, which was an article in Nature magazine, got a lot of media attention. It really sort of sets the stage for a, proceeding towards a clinical use. And I think that's, it really does kind of open the door, because what they showed importantly in that paper was that there were very few off-target editing events that happened, and also they could avoid something called mosaicism, which means editing happening in just some of the cells of the resulting developing embryo. And so we now understand that when applied in certain ways, this kind of technology can be very robust in viable human embryos. So it really does, we can no longer say, well, there's still a lot of technology development to be done before we have to worry about that application. It's now a question of, we know this can work, are we really going to go there or not?
Thanks again to Professor Doudna for that.